This is a genome of the SARS-CoV-2 virus, aka the virus that causes COVID-19, aka the coronavirus. It's almost 30,000 characters long and contains all the instructions to make proteins through these 4 bases in the form of RNA. I'm especially concerned with how we can read the bases from such a thin molecule so quickly, yet be very accurate. So let's find out.
Before, we used a technique called Sanger sequencing, but that was expensive and lame. So Alexa, which Illumina later bought, invented a cool new method at a tiny fraction of the cost called Next Generation Sequencing, or NGS. Our first step is sample preparation, so we need to get a sample of the virus from an infected person. And since SARS-CoV-2 uses RNA, we translate it into DNA, which uses T instead of U. We can't read all 30,000 bases at once, so we cut into smaller pieces of like 300 to 600 called reads. To put them in the correct order, we attach index sequences in the form of extra bases. We then add even more bases to the end, which will be needed later when we put the strands in a sequencing machine.
But that's just sample preparation. How do we actually sequence it?
First, we choose one of the many machines Illumina has to offer, let's say the MySeq, which specializes in small genomes like viruses, grab its flow cell where the sequencing will be done, and introduce the reads. Each flow cell contains short pieces of genetic material called oligos, which will attach to the strand at either side because of the extra bases we put at the ends during sample prep. The original strand is copied and washed away. The second strand then attaches to another oligo, like a bridge. This is repeated thousands of times through bridge amplification to create one cluster which all have the exact same sequence of bases, along with millions of other clusters on the flow cell, each corresponding to a different read. We then use sequencing by synthesis, whereby many bases with unique fluorescent markers for each base are introduced into the system. They then emit a light when they attach to the strand, and the colors are recorded in many different pictures where we can determine the sequence of each read by the color of the cluster. We then do the same for the index sequence we placed during the sample preparation to help order the reads. Although this process is very accurate, there are still several mistakes in a 30,000 base sequence, so the genome is read many times, sometimes thousands of times, to iron out those mistakes. Finally, we use a computer algorithm which takes out the mistakes and overlapping parts of the read to generate one single sequence of characters called the genome.
That's really awesome, but why should we care? I actually asked researchers and professors from Harvard and the UC system, and here's their response.
Yeah, that's a really great question. Knowing the genome of the virus tells us a lot of things. Trying to understand how the virus is potentially changing in people.
If there could be some cross-protection between SARS-CoV-2 and other coronaviruses.
How we can potentially target and treat the virus with drugs that are available.
Genome sequencing is crucial to help fight the virus and end the suffering of so many, and will continue to in the future. I think it's a great example of the immense good technology can bring, and it gives me hope that we can do great things. Stay happy and healthy.